Hello everyone. As we know, immune system is composed of immune organs, immunocytes, and immune molecules. We have introduced immunocytes. In the following two sections, we will talk about immune molecules. You may ask, which kind of immune molecules take part in immune defense? What are the rules of immune molecules in immune response to pathogens? To make clear these questions, we are going to introduce antibody, complement, cytokine, and MHC briefly. The first molecule is antibody. Antibodies are circulating proteins that are produced in vertebrates in response to exposure to foreign structures known as antigens. All antibodies belong to immunoglobulins. Antibodies are the mediators of human immunity against all classes of microbes. We only introduce the structure and functions of antibodies. All antibody molecules share the same basic structural characteristics, but display remarkable variability in the regions that bind antigens. An antibody molecule has a symmetric core structure composed of two identical light chains and two identical heavy chains. Both heavy chains and light chains consist of amino-terminal variable V regions that participate in antigen recognition and carboxyl terminal constituent C regions. So, every antibody molecule has at least two antigen binding sites. According to the differences of antigens, immunoglobulins can be divided into 5 classes or S-types. There are 5 classes of antigens, including gamma, alpha, mu, delta, and epsilon. Therefore, there are 5 classes of immunoglobulins, including IgG, IgA, IgM, IgD, and IgE. Also, according to the differences of L chain, immunoglobulins can be divided into 2 types, and there are 2 types of L chain, kappa, lambda. Most of sequence differences and variability among different antibodies are confined to 3 short stretches in the V region of the heavy chain and 2-3 stretches in the V region of the light chain. These segments of the greatest diversity are known as hypervariable regions, HVR. Except HVR, the other regions of V region are relatively conserved and called framework region. Because HVR sequences form a surface that is complementary to 3-dimensional shape of the bound antigen, so the hypervariable regions are also called complementarity determining regions, CDR. Proceeding from either the VL or the VH amino terminals, these regions are called CDR1, CDR2, and CDR3. Then you may ask how antibodies perform their functions. Generally, antibodies function in 4 main ways: neutralization, opsonization, complement fixation, and ADCC. All of the functions of antibodies are dependent on their ability to specifically bind antigens. The main functions of antibodies are to neutralize and eliminate infectious microbes and microbial toxins. Many microbes enter host cells by the binding of particular microbial surface molecules to membrane proteins or lipids on the surface of host cells. Antibodies would block the binding of these microbes and toxins to cellular receptors. Neutralization is mediated entirely by binding of antigen and does not require participation of the immunoglobulin constituent regions. Usually, some microbes are resistant to B phagocytosis. IgG or IgM attaches to the surface of a microbe by Fab and binds to the surface receptors of neutrophils or microbes by Fc simultaneously. So, antibodies act as a bridge between microbes and phagocytes, which increases the efficiency of phagocytosis and the killing ability of phagocytes to microbes. When antibody binds to target cells by Fab and its Fc binds to effector cells such as NK cell, macrophages, or neutrophils, which will lead to activation of effector cells and result in lysis of the antibody-coated target cells, which is called antibody-dependent cell-mediated cytotoxicity, ADCC. Usually, IgM, IgG1, 2, 3 activate complement by classical pathway. IgA, IgG4, IgE activate complement by alternative pathway. Finally, MAC is formed to lyse the target cells. Next, we would introduce complement. Complement is a system of serum and cell surface proteins, including more than 30 proteins, that interact with one another and with other molecules of the immune system to generate important effectors of innate and adaptive immune response. The soluble proteins of the complement system are synthesized in the liver and are secreted as non-active forms called zymogens. The cleavage is required for complement activation. The cleavage of a zymogen usually produces a larger active fragment of enzymatic activity and a small fragment with inflammatory effects. Complement can be activated by a classical pathway, alternative pathway, or MBL pathway. Finally, 3 pathways enter the terminal pathway, and MAC is formed. Classical pathway is initiated by antibody-antigen complex. After binding to antigen, the configuration of antibody is changed to exposure to the complement binding site to activate complement by a cascade reaction. In this pathway, C3 convertase is C4b to A, and C5 convertase is C4b to A3b. In the alternative pathway, C3 convertase is C3b BB, and C5 convertase is C3bN BB. In this pathway, complement can recognize self from non-self. In addition, alternative pathway is important in large mechanism of complement activation. The MBL and C reactive protein are produced by liver after microbe infection, MBL binds to mannose residues on polysaccharide or microbe to activate MASP. MASP is similar to C1R and C1S, and MASP activates C2, C4, which is similar to classical pathway. The late steps of complement activation is formation of membrane attack complex, MAC. MAC is the lytic complex of the terminal components of the complement cascade, including C5, 6, 7, 8, and multiple copies of C9 that form pores in the membrane of target cells. The MAC causes lethal ionic and osmotic changes in cells. Now let's introduce the biological functions of complement. Complement activation by classical pathway, MBL pathway, and alternative pathway leads to the formation of MAC. MAC mediates the lysis of target cells. Similar to antibody-mediated opsonization, C3b and 4b, in the surface of microorganisms attached to complement receptor CR1 on phagocytes and promote phagocytosis of microbes. Antigen-antibody C3b complex can adhere to the C3b receptor, which exists on the surface of RBC or platelet. This facilitates phagocytosis of immune complex by phagocytes. After activation, cleaved complement C3a, C4a, and C5a act as anaphylaxis by binding to receptors on mast cells to induce inflammatory reaction. In addition, C3a and C5a mediate chemokine-like action, while C2b and C4a mediate kidney-like action. Finally, complement mediates immune regulation by helping APC to present antigen, promoting the proliferation and differentiation of B cell, or enhancing ADCC induced by CR1. Therefore, complement can be activated by classical pathway, MBL pathway, or alternative pathway. Activated complement mediates lysis of bacteria and cells, opsonization, elimination of immune complex, inflammatory reaction, and immune regulation. In this section, you should know the structure and functions of antibodies, 3 activation pathway, and biological functions of complement. Thanks for your watching.